Chapter 19 Harvesting Electrons from the Cycle. In Chapter 18, we learned how glucose is metabolized to acetyl-CoA. This reaction, catalyzed by the pyruvate dehydrogenase complex, is the irreversible link between glycolysis and the citric acid cycle. Here we're going to learn a common fate of acetyl-CoA when oxygen is present and energy is required. It's a complete combustion of the acetyl group by the citric acid cycle. The citric acid cycle is the biochemical hub of the cell, oxidizing carbon fuel, usually in the form of acetyl-CoA, and serving as a source of precursors for biosynthesis. Let's look at the big picture again. Where is this chapter 19? It's this area. Here's the outline of this chapter. We first talk about the citric acid cycle consists 2 stages. Then we talk about stage 1, then stage 2, and then we talk about how this pathway is regulated, and then end with a case study. The citric acid cycle oxidizes the acetyl fragment of the acetyl-CoA to carbon dioxide. It's a complete combustion. In the process of this oxidation, high energy electrons are captured in the form of NADH and FADH2. The key function of the citric acid cycle is to harvest these high energy electrons from the carbon fuels. This is total net reaction of the citric acid cycle. The citric acid cycle consists of 2 stages. The first stage, a 2-carbon acetyl group condensed with a 4-carbon oxaloacetate form a 6-carbon compound citrate. The 6-carbon citrate undergoes 2 oxidative decarboxylation, generate 2 molecules of carbon dioxide. This is the stage 1. In stage 2, the oxaloacetate is regenerated. Both stages generate high energy electrons in the form of NADH, FADH2, and also synthesize ATP. Let's look at another big picture. Chapter 19 is about the citric acid cycle. Acetyl-CoA, mostly from glucose, may come from fatty acid and amino acids, enter the citric acid cycle, produce 8 electrons. In chapter 20, we're going to learn these electrons transfer through electron transport chain, reduce oxygen, and produce the proton gradients, which generate ATP. The 2 pathway enters in the inner mitochondrial membrane, and they are part of a cellular respiration. Let's look at the stage 1, step 1. The acetyl unit carried by acetyl-CoA entered the citric acid cycle through here. It condenses with oxaloacetate, forms citrate. The reaction is catalyzed by a synthase. Synthase is an enzyme that catalyzes a synthetic reaction in which two units are joined, usually without the direct participation of ATP. You can see here the 2-carbon acetyl unit carried by acetyl-CoA joined with 4-carbon oxaloacetate form citrate, 6-carbon. This is a citric acid cycle, or called Krebs cycle, or tricarboxylic acid cycle. It totals 8 steps, involve 8 enzymes. Since we already watched the YouTube video and learned every step of it, I'm not going to explain every step. This is a summary of the 8 steps, including the reactant, the product, and the enzyme involved in each reactions, and a detailed outcome. This is the net reaction of the citric acid cycle. We can see 1 acetyl-CoA will produce 3 NADH and 1 FADH2 and 2 carbon dioxide, 1 ATP. The citric acid pathway is regulated at several points. The pathway is regulated primarily by the concentration of ATP and NADH. The control points are the enzyme isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. The control of citric acid cycle. The citric acid cycle is regulated primarily by the concentration of ATP and NADH. We recall that the pyruvate to acetyl-CoA is inhibited by the ATP and NADH and stimulated by ADP and pyruvate. In citric acid cycle, there are 2 control points. First one is from isocitrate to alpha ketoglutarate. The other one is from alpha ketoglutarate to succinyl-CoA. Both control points are inhibited by ATP and NADH, which means when the body has high ATP, when ATP is not needed, then the citric acid cycle will be inhibited, will be reduced. Let's see where the 2 control points in the big detailed map. You can see it's from here, the isocitrate dehydrogenase, and this one is alpha ketoglutarate dehydrogenase complex. Both enzymes are key control points, and both reactions have NAD plus and NADH involved. The citric acid cycle is a source of biosynthetic precursors. Many of the components of the citric acid cycle are precursors for biosynthesis of key biomolecules. We can see that the oxaloacetate can synthesize glucose through gluconeogenesis, and all the other arrows can see that those intermediates can be used to synthesize other compounds. The citric acid cycle must be capable of being rapidly replenished. The cycle is crucial for generating biological energy and is also a source of building blocks for biosynthesis. Therefore, must be replenished if any are drawn off for biosynthesis. The key intermediate oxaloacetate is formed by the carboxylation of pyruvate in a reaction catalyzed by the biotin-dependent enzyme pyruvate carboxylase. We learned that before. Pyruvate carboxylase replenish the citric acid cycle. The rate of the citric acid cycle increases during exercise, requires the replenishment of oxaloacetate and acetyl-CoA. The oxaloacetate can be replenished by formed from pyruvate catalyzed by the pyruvate carboxylase. And acetyl-CoA can be formed from pyruvate or fatty acid. This is a clinical insight. Defects in citric acid cycle contribute to the development of cancer. The defects in succinate dehydrogenase, fumarate isocitrate dehydrogenase, or pyruvate dehydrogenase kinase can contribute to the development of cancer. A mutant isocitrate dehydrogenase can alter gene expression. Other defects contribute to the use of the aerobic glycolysis by cancer cells.